Hi students, this is A. Mandri Kumar Rajesh. In today's video, I am going to teach you Trihybrid Cross with the help of an important method called Forkline method. Forkline method. First, I give you the definition for Trihybrid Cross. It is a cross between two individuals which is made to study the inheritance of three pairs of alleles belonging to three different genes. So, it is a cross between two individuals which is made to study the inheritance of three pairs of alleles belonging to three different genes. So, this is the definition for trihybrid cross. Then, why I am telling the Forkline method is an important method? Because, for instance, we are analyzing a cross involving three genes. These three genes can produce 8 gametes in each individual. These three genes can produce 8 gametes in each individual. So, it is a male. It can produce 8 gametes. So, it is a female. It can produce 8 gametes. Results in the young ones, we can obtain 64 young ones. That means in Punnett square, we have to draw 64 boxes. Then 8 plus 8, 16 grids. In Punnett square, we have to draw 16 grids and 64 boxes. Suppose we are analyzing a cross involving 4 genes. So, these 4 genes can produce 16 gametes in each individual. So, we have to draw 16 plus 16, 32 grids. Then results in the young ones, 256 young ones we can obtain by this cross. So, in Punnett square, we have to draw 256 boxes, then 32 grids. So, it is a complicated, tedious, extremely a difficult process to manually enter each genotype. For that type of complicated process, we can make use of Forkline method. Now, I am going to tell you how to prepare a Forkline diagram for a trihybrid cross. For making a Forkline diagram for a trihybrid cross, we have to make a cross between two individuals which contains 3 types of characters. That means it must contain 3 types of genes. So, the phenotype of homozygous dominant is tall, yellow, round. So, this is the phenotype for homozygous dominant parent. Cross with homozygous recessive. Now, I am going to write the phenotype for homozygous recessive parent. Dwarf, green, wrinkled. So, this is the phenotype for the recessive parent. Now, I am going to write the genotype for the dominant one. So capital T, capital T, capital Y, capital Y, capital R, capital R. cross with the genotype of the recessive is small t, small y, small r, small r. Then the gametes produced by the dominant one is capital T, capital Y, capital R, and the gametes produced by the recessive one is small t, small y, small r. Then fusion of gametes takes place, results in the development of F1 generation. In F1 generation, we could observe all the young one possess heterozygous dominant character. That means the genotype of the young one is capital T, capital Y, small y, capital R, small r. So in F1 generation, we could observe all the young ones possess heterozygous dominant character, and it shows the phenotypic character tall, yellow, round. Then this F1 generation is allowed to self-pollination. So F1 cross with F1, that means capital T, capital Y, small y, capital R, small r, cross with capital T, capital Y, small y, capital R, small r. Up to this, we can follow the same procedure what we have followed in monohybrid or dihybrid cross. Hereafter, we are going to follow the different rules and regulations. So now I am going to figure out forkline diagram for trihybrid cross. For making a forkline diagram, first we have to do monohybrid cross between these two individuals for each characters. That means we have to do monohybrid for height character, seed color, seed shape. So now I am going to draw Punnett square for each characters. First I am going to draw the Punnett square for height character. This is for the seed color character, and this is for seed shape. Now we can place the gametes in respective boxes. So capital T, capital T, small t. I am taking the gametes from between these two. I am taking gametes between these two individuals, capital T, capital T, capital T, capital T. So results in capital T, homozygous dominant, tall. Capital T, small t results in heterozygous, tall. Capital T, small t results in heterozygous, tall. Small t results in homozygous, dwarf. Then for the seed color, capital Y, small y, capital Y, small y, capital Y results in yellow color, homozygous dominant. Capital Y, small y results in yellow color, heterozygous dominant. Capital Y, small y, heterozygous dominant, yellow in color. Small y, homozygous recessive, then young one will be green in color. Then for the seed shape, capital R, small r, capital R, small r, capital R, it shows round, homozygous dominant. Capital R, small r, heterozygous dominant, then it shows round character. Capital R, small r, it shows heterozygous dominant, then character will be round. Small r, it is a homozygous recessive one, so wrinkled, it shows wrinkled character. Then the genotypic ratio for each character is 1 is to 2 is to 1, 1 is to 2 is to 1, 1 is to 2 is to 1. Now I am going to write the possibilities. Here we have capital T, only one young one. So here we have homozygous dominant, only one, heterozygous dominant, 2, homozygous recessive, 1. Then for this, homozygous dominant is only 1, capital Y, heterozygous dominant is capital Y, small y, 2 numbers. For homozygous recessive, only 1, small y, small y. For this homozygous dominant is only 1. For this, 2 numbers are present, they are capital R, small r. For this only 1, the recessive character is only 1. Now it can be written as 1 by 4, 2 by 4, 1 by 4, because among the 4 individuals the homozygous dominant is only 1. Among the 4 individuals the heterozygous dominant is only 2. Among the 4 individuals the homozygous recessive is only 1. So we can written as 1 by 4, 2 by 4, 1 by 4. And also we can written for the other phenotypic ratio also, 1 by 4, 2 by 4, 1 by 4, by 4, 2 by 4, 1 by 4. Now I am going to create Forkline diagram with the help of an genes being considered. First I am going to take the first possibility in height character. First I am going to take the first possibility in height character, that is capital T in the proportion 1 by 4. So 1 by 4 capital T. Now I am going to take the results from the second square. Now I am going to take all the results from the second square, that is seed color. Here we have 3 possibilities in different proportions. Here we have 3 possibilities in different proportions. So we have to draw 3 fork. We have to draw 3 fork. We have to draw 3 fork. Now I am going to place all the possibilities with their proportion. 1 by 4 capital Y, 2 by 4 capital Y, small y, 1 by 4 small y. Now I am going to take all the results from the third square. Now I am going to take all the results from the third square, that means the seed shape. So here we have 3 possibilities with 3 different proportions. Here we have 3 possibilities with 3 different proportions. So we have to draw 3 fork lines for this character. That results are going to attached with the seed color. So now I am going to rub this. Now I am going to place all the possibilities with their proportion. 1 by 4 capital R, 2 by 4 capital R, small r, 1 by 4 small r. Likewise, I am going to write the results for this fork line also. So we have to draw 3 forks. 1 by 4 capital R, 2 by 4 capital R, small r, 1 by 4 small r. Likewise for this seed shape also, seed color also. I am going to attach the value from the third square with the seed color. So again we have to draw 3 Forkline. 1 by 4 capital R 2 by 4 capital R small r 1 by 4 small r. Why we have to plot all the genes being considered? Because each gene can assort independently. Why we have to plot all the genes being considered? Because each gene can assort independently. So we have to plot all the possibilities with their proportion. After this, we have to multiply the values along each Forkline pathway. To get F2 offsprings probability of phenotypic and genotypic ratio. Now I am going to multiply each Forkline pathway. First I am going to multiply this Forkline pathway. That is 1 by 4 capital T multiply with 1 by 4 capital Y multiply with 1 by 4 capital R results in 1 by 64 capital T capital Y capital R is 2. Then I am going to cross this value with this value, then this value. So the result is 1 by 4 2 by 4. So 2 by 64 capital T capital Y capital R small r capital T capital Y capital R small r. Then I am going to multiply this value with this again with this value. So it results in 1 by 64 capital T capital Y small r. Likewise, I am going to multiply this value with this value. So 1 by 4, here 2 by 4, here 1 by 4. So in total 2 by 64 capital T capital Y small y capital R. Then I am going to multiply this value with this value and this value. So here we have 2 by 4, here 1 by 4. So total 4 by 64. So capital T capital Y small y, then capital R small r. Then the third possibility 1 by 4, 2 by 4 and 1 by 4. So 2 by 64 capital T capital Y small r capital T capital Y small r. Likewise, we are going to multiply this value with this value. So here we have 1 by 4, here we have 1 by 4, here we have 1 by 4, results in 1 by 64 capital T small y capital R. Then this value is going to multiply with this value, then this value. So here we have 2. So 2 by 64, capital T, small y, capital R, small r. Then the last one is capital T, small y, small r. So it results in 1 by 64, capital T, small y, small r. So we have multiply all Forkline pathway to get F2 offsprings in the probabilities of phenotypic ratio and genotypic ratio. Likewise, I am going to multiply for the second possibility, that is capital T, small t, then the third one is small t. So please stay with me. You can feel somewhat hectic, so don't worry, it will be easier for you when you practiced. So please stay with me, don't get bored. Okay, so please stay with me. Now I have written all the results of 3 Forkline diagram. I have written all the results of 3 Forkline diagram. For this I have shown how to prepare a Forkline diagram. For this too, I have simply written the results. Here you can find 64 young ones. Please count that. You can find 64 young ones with their genotypes. Here you can find 64 young ones with their genotypes. Now I am going to write the phenotypic ratio of this trihybrid cross. So you can see here the phenotypic ratio of trihybrid cross. The ratio is 27:9:9:9:3:3:3:1. So that's all about trihybrid cross. So we can use Forkline method to figure out the probability of genotype and phenotype for any kind of crosses, for any kind of polycrosses. It will be a reliable, user-friendly method to figure out any kind of crosses. With this information, I am going to conclude this video. I hope this information will be helpful for you. Please stay safe, stay home, and thank you for watching this video. Thank you.